everyone's having a good day. My name is Jacob Mapp. I'm going to be a senior in biomedical engineering, and right now I'm in the Aspen Wall Lab. Okay, so my research is on capillary electrophoresis. So what it is basically an analytical chemistry tool that allows you to separate and analyze different components within a given sample of study. So the way it works is, as you can see, there's a capillary with a capillary window. It has a laser and then a detector. So basically, there's an anode and a cathode dipped in each of the vials that the capillaries dip in. Once you turn on a voltage supply, it causes an electric field to occur within the capillary. What that does is it pulls all of the components within the capillary from the anode towards the cathode. And as this occurs, if you have any fluorescent dye inside, as the dye reaches the capillary window, the laser excites that dye, and that dye emits its own color. In our case, we're using fluorescein, which emits about 525 nanometers well, the light is about 525 nanometers. And so that is the detector, and we can just see that as a signal on the computer. So that's the normal, classic, conventional setup. So the problem with conventional CE instrumentation, or CE is capillary electrophoresis for short, is that it's expensive, it's large and bulky, it's not mobile, and it's also very complex, so it's not very user-friendly. So my project is, I can pass this around, so basically what you see up there is this. It's about the size of a computer mouse, which is pretty cool because, well, I'll explain the parts of it. So basically, as you can see here, it takes up all of the classic components of CE and compacts it. So right here you have a laser that shoots through here. We have a dichroic mirror that reflects blue light. Goes through a microscope objective right onto the capillary window. So as the fluorescein reaches the capillary window, it excites it, and the fluorescein emits its green light. The green light passes through the dichroic mirror and past the filter and into the detector. Okay, so why does all this matter? So one, it's quick, which allows for quicker patient diagnosis. Two, it's mobile, which would allow for on-site testing. For instance, if you're not in necessarily a hospital setting, you're more in, out in the field, because not only can this be applied to the medical field, it can be applied to, say, water testing as well, to check for bacteria. So as well as it's cheaper, obviously. I printed that in the library for about $5, which is great. A lot less than $5,000 to $10,000 for the conventional capillary electrophoresis machines. It's also smaller, as you can see. You can put that as well as all the necessary components, such as a computer and voltage supply, in a backpack. Also, it's easier to use. It's pretty simple. So in areas where they need something like this but don't have the trained medical personnel, it can be, taught to use easier. Okay, so how well is it working? Up here we have a graph of 3 different runs that I did using that specifically. And so this is done with 5 nanomolar concentration of fluorescein. And so as we can see, the migration times are within about 10 to 15 seconds of each other. And so this is important because this is the migration times that we need to be consistent in order for this to be a viable option for a cheap and mobile way of performing capillary electrophoresis. So as of now, it's pretty consistent and reproducible. And yeah. Okay, so how my instrument goes from research lab to hospital or out in the field. So let's say you have a disease, doctor needs some help figuring out what you have. So here's how to use it. First, you take a sample of blood or some other biological sample. You dilute it and you put in fluorescein label antibodies. And so fluorescein is, like I was saying earlier, it's a fluorescent dye that gets excited by bluish light, so 450 around light, and it emits 525 nanometer light. So then if there is any bacteria in something, whatever they took, let's say blood, if there's bacteria, these antibodies that have fluorescein attached can attach to those bacteria cells. And so what happens is you run it through this. So this is basically what I passed around, but all connected. That's the capillary right there, and it has the laser shooting through it, as you can see. So if you run it through, then you will see a peak, which is what we saw before on the graph. You see a peak, which indicates that there's fluorescein, and in this case, it'll indicate that it has attached to a bacterial cell, indicating that you do, in fact, have what you're checking for. And then you can apply the correct treatment based off of that. So And so recap, this allows for quick but high resolution separation. And my project is trying to work on making it smaller, more compact, easier to use, all the above. And so the current goal is to make it even more consistent, more reproducible, and potentially go down to less concentrations than what I'm using right now. So yeah, if there are any questions, let me know. Also, I'd like to give a thank you to Hubert as well as Dr. Aspinwall. Yeah. Thank you.